Inside every one of your cells sits a sequence of 3 billion unique chemical letters. Even at this scale, changing just a single letter creates what is known as a single nucleotide variant, or SNV. This microscopic typo is responsible for 50 to 90% of all known pathogenic genetic variants in humans. Devastating conditions like sickle cell and beta thalassemia are the direct consequences of a single wrong base pair. In the human genome, one single letter marks the difference between a healthy life and a debilitating genetic disease. Traditional CRISPR-Cas9 was the first tool built to manipulate our DNA, but its mechanism is essentially that of molecular scissors. To fix a mutation, traditional CRISPR completely severs both strands of the DNA double helix. The cell then uses an error-prone emergency repair mechanism to fuse the broken strands back together. This often backfires, creating unintended mutations, insertions or deletions, known as indels at the repair site. While scissors are effective for cutting, fixing a single letter typo requires the precision of an eraser and a pencil. Shattering the DNA backbone to fix one letter creates too much collateral damage to be a safe option for many patients. Base editing, introduced in 2016, provides a search and write solution that avoids breaking the DNA backbone. The editor uses a modified CRISPR-Cas9 nickase protein fused directly to an enzyme called a deaminase. A guide RNA navigates the editor to the mutation and unzips the DNA, but it does so without severing the structural rails. The deaminase then performs chemical surgery, directly converting a cytosine base into a uracil. To make the change stick, the editor nicks the opposite, unedited strand, which tricks the cell into treating the old strand as the one that needs repair. The cell repairs that nick by inserting an adenine to match the new uracil, effectively converting a CG pair into a TA pair. This allows scientists to edit the sequence through direct chemical conversion, bypassing the risks of shattering the genome. Early versions of this tool sometimes converted adjacent, unintended bases, a problem known as the bystander effect. Scientists solved this by engineering smaller enzymes that restrict the editing window to a single target base. These improvements led to the development of adenine base editors, which can also fix A to G mutations. This iterative engineering has transformed base editing from a laboratory theory into a precise clinical instrument. Base editing technology has now successfully moved beyond laboratory research and into the bodies of living human patients. In recent trials, the drug CS101 used base editing to successfully treat patients with severe beta thalassemia. Verve Therapeutics has also seen success using adenine base editors to target the PCSK9 gene in patients with cardiovascular disease. This science has progressed far beyond the initial Human Genome Project, when researchers were focused primarily on deciphering the sequence. Base editing demonstrates that the future of medicine lies in the ability to fix genetic errors at their source, correcting the genome one letter at a time.